Here we'll learn an overview of viruses with a focus on their general morphology. Start a table and denote that viruses are obligate intracellular parasites because they rely on host cellular energy and proteins for their own replication. Indicate some key features of virion morphology. The nucleocapsid comprises the viral genome, which consists of either DNA or RNA surrounded by a capsid. Denote that the capsid is a protein coat that protects the genome. Indicate that some virions are enclosed by an envelope which is made of lipids, proteins, and glycoproteins. Lastly, denote that virions come in a range of sizes from 20 to 400 nanometers. Let's begin with the nucleic acids that comprise the viral genome, which can be linear or circular. First, indicate that virions with DNA can have single or double-stranded DNA. Most RNA viruses are single-stranded and are classified according to the polarity of their RNA. Indicate that they can be positive sense, which means that the RNA is like messenger RNA, or they can be negative sense, which means that they are like templates for messenger RNA that have to be transcribed into positive RNA. Be aware that there are exceptions and complications to this classification system, which we'll learn about more in our tutorial on virus replication. Next, let's consider some common symmetrical capsid shapes: helical, icosahedral, and complex. First, show a helical capsid. We use filovirus as our example. Notice that the viral genome is enclosed by the helical capsid proteins. Furthermore, indicate that this virus is enclosed in an outer envelope, which we'll learn about more in a moment. Next, show an icosahedral capsid. We use adenovirus as an example. To see its inner structure, we cut it in half. In reality, this virus comprises 20 equilateral triangles that form a shell around the genome. The viral genome resides inside the capsid, and viral attachment proteins project from the corners of the capsid triangles. Finally, let's show an example of a complex capsid. We use bacteriophages as our example. Show that the bacteriophage comprises an icosahedral head enclosing the viral genome and a tail that facilitates delivery of the viral genome into the bacterium. Next, write that the virions can be naked or enveloped. An important example of an enveloped virus is influenza A. Write that the envelope is acquired from the host cell during viral budding. First, show the outer viral envelope and the inner nucleocapsid, which comprises the capsid and viral genome. On the surface of the viral envelope, show glycoproteins. These projections facilitate entry into host cells and stimulate the immune process. Let's compare naked capsid virions and enveloped capsid virions. Write that naked viruses are relatively stable. They are resistant to temperature changes, acids and proteases, detergents, and drying. Thus, they can survive outside the host and are spread easily, for example, via the fecal-oral route. Enveloped capsids are more fragile. They're labile to heat, acids, detergents, and drying. Thus, transmission is often via respiratory or sexual routes. With this information as a backdrop, let's learn about key families of DNA and RNA viruses. We'll begin with the DNA viruses. First, we indicate examples of naked DNA viruses. Next, enveloped DNA viruses. Now indicate that most DNA viruses are double-stranded. Parvoviridae is the single-stranded exception. Most DNA viruses have icosahedral symmetry. Poxviridae has a complex shape, and it's the exception. And most DNA viruses replicate within the host nucleus. Poxviridae, again, is the exception. It replicates in the cytoplasm. Now remind ourselves that RNA viruses can be positive-stranded, negative-stranded, or ambisense, which means it contains both positive-sense and negative-sense RNA. So now list examples of the following: naked positive-stranded RNA viruses, enveloped positive-stranded RNA viruses, and negative-stranded RNA viruses that are enveloped. Indicate that Reoviridae is a double-stranded, segmented RNA virus with a double capsid. Write that most RNA viruses are single-stranded, but as we just noted, Reoviridae is the exception. Many RNA viruses display helical symmetry. We list out several exceptions. And most RNA viruses replicate in the host cytoplasm, indicate that Retroviridae and Orthomyxoviridae are the exceptions because they replicate in the host nucleus. Before we conclude, let's draw some simple variants to summarize key points regarding virus morphology. We'll start with naked, also known as non-enveloped capsids, show that they can be in helical arrangement, so that the nucleic acids are wrapped in capsid proteins to form a helical shape. Or the capsid can be icosahedral, with the nucleic acids in the center and viral attachment proteins projecting from the corners. Several of the DNA viruses have this morphology. Finally, show that the enveloped capsids are similar, but with an additional outer layer of lipids and glycoproteins. Draw some representative envelope glycoproteins on their surfaces. This concludes our diagram.